good afternoon to our distinguished audience in China, and a very warm welcome to our international participants joining from around the world. That's a very difficult word to most people in China. So I say that again, around the world, okay. We are truly delighted to have you with us for today's seminar on ocean carbon sequestration and climate resilience, an event held as part of an ongoing series endorsed by the UN Ocean Decade and sponsored by the Global Ocean Negative Carbon Emissions ONCE program, led by Professor Yongyu Zhang from Xiamen University. I am Chuanlun Jiang from the Southern University of Science and Technology in Shenzhen. Joining me as co-host is Dr. Yernar Sailybayev from the Caspian Environmental Consortium in Kazakhstan. Kazakhstan. Okay. It is our great honor to introduce today's keynote speaker, Professor Yongyu Zhang, Director of the Research Center for Marine Biology and Carbon Sequestration at the Qingdao Institute of Bioenergy and Bioprocess Technology, Chinese Academy of Sciences. Professor Zhang is a leading expert in biologically driven carbon sequestration with groundbreaking work on seaweed farming. His a recent paper is in the very final stage, submitted to Nature Communications, as I learned. So Professor Zhang has published extensively in top-tier journals, and his research has enhanced our understanding of marine carbon storage. He will be presenting today on the topic, significant potential of seaweed farming in mitigating climate change. So I will also introduce today's panel members. After Professor Zhang's presentation, we'll be joined by three outstanding panelists for an insightful discussion on today's topic. Professor Guang, if you can let us know you are here. Yeah, okay. Professor Gao is from Xiamen University. He's an internationally recognized scholar in algae-based carbon capture, utilization, and storage, and a contributor to major UN environmental assessments. We have Professor Zengjie Jiang from the Yellow Sea Fisheries Research Institute, Chinese Academy of Fishery Sciences. Professor Jiang is an expert in sustainable marine culture and climate aquaculture interactions, and a chief scientist in the National Shellfish Industry Technology System. We also have Professor Xiao from Zhejiang University, recipient of the Keeling Curve Prize and a leading researcher in aquatic ecological restoration and carbon capture technologies. Each of these scholars has made exceptional contributions to their fields. For more details on their research and accomplishments, I will invite you to visit the Global ONCE website at www.global-once.org. Without further ado, let's extend a warm welcome to Professor Yongyu Zhang, Professor Zengjie Jiang, and Professor Xiao. Okay, Yongyu, now the floor is yours. You have 30 minutes for your presentation.
Firstly, I need to thank Professor Zhang to invite me here. It is a good opportunity for me to introduce our work. today my prediction focus on the seaweed. Wait a minute, seaweed. My prediction will focus on the significant potential of seaweed farming in mitigating climate change. As we know that the ocean is the largest carbon sink on the Earth. Its carbon pool is 50 times than that of the atmosphere and 20 times of that of the Earth. Every year, 30% of the CO2 emission from the by humans will be absorbed by the ocean. Therefore, the ocean play an important role in regulating the climate change, regulating the climate. So far, the well-known mechanisms driving ocean carbon sequestration mainly refer to 4 pumps. This is a vivid description. It is the biological carbon pump, the microbial carbon pump, and the solubility carbon pump, and the CCP, that is carbonate count pump. I think everybody here have a good understanding of the details mechanism of the 4 pumps. So I would not take more time to introduce in details. Here I want to emphasize that phytoplankton or macroalgae play a very important role underpin these 4 processes. For example, the biological carbon pump and the CCP pump, they are dominated by the phytoplankton. And for the microbial carbon pump, we know that the macroalgae or the phytoplankton provide the basic raw materials or organic carbon to support the MCP processes. And for the CCP, the microbial also driven the carbon, the CO2 absorption by photosynthesis. This is occurred in the open ocean. However, in the coastal ocean, macroalgae, or called seaweed, are the main primary producers in coastal oceans. It is reported that their net primary productivity even exceed the total contribution by the well-known blue carbon ecosystems that include the mangrove, salt marsh, and seagrass bed. There are more than 12,000 different species of macroalgae worldwide. This includes the cultured seaweed or macroalgae and the more diverse wild seaweed. Under climate change scenarios, in order to achieve the goal of carbon neutrality, in recent years, seaweed farming has received wider attention. However, at present, how to close this? However, at present, the carbon sequestration mechanism and the efficiency are not yet well understood. Let me give a very simple example. There is a big difference in the carbon sequestration effect between the farmed and wild seaweed. For the wild seaweed, they grow and die in the ocean. Therefore, their biomass play a very important role in carbon sequestration. While for the carotid or carotid seaweed, since once material, the biomass will be harvested and mostly was used as food. Therefore, the biomass carbon can return back to the atmosphere rapidly. Actually, the definition of carbon sink or carbon sequestration has a time scale convention. That means that only the carbon can be isolated from the atmosphere for a long term, at least more than 100 years. It can be regarded as the real carbon sequestration. In the past, our previous work mostly focused on the wild seaweed. It is different that the wild seaweed usually grow on a rocky substrate. So the microalgae derived carbon is rarely absorbed in the sediment, or it is very difficult to be buried in the sediment. Therefore, in the past, the wild seaweed was considered to have no carbon sequestration effect. In recent years, the scientists find that most of the seaweed carbon, the wild seaweed carbon, was exported outside, and many of them are exported to the deep ocean. So in order to track the long-distance export of microalgae fragment or particles, the scientists by using the eDNA or by examining the microalgae eDNA, they do a global investigation. They find that the microalgae POC can be exported as far as 5,000 kilometers far away and can reach the deep ocean at depths of 4,000 depths. Here, I want to mention that here, this just refers to the POC, not the DOC, since the DOC is very difficult to extract its DNA. Therefore, the export of microalgae-derived DOC remains unclear and continues to be a great challenge. Consider that the biomass of seaweed play a vital role in carbon sequestration. In recent years, a new strategy named deep sea seaweed sinking strategy, that means cultivate seaweed and then sinking them into the deep ocean, was proposed as a strategy to enhance carbon sequestration. However, this strategy is now still controversial. On one hand, we know that sinking the algae to the deep ocean is a waste of resources, as microalgae is highly nutritious and can be used as food and other high-value products. On the other hand, that no corresponding research has been conducted. We don't know what will happen or what's the potential ecological risks. So now this strategy is still controversial, has not been conducted yet. In contrast, the farmed seaweed, earlier I have mentioned the wild seaweed has very few carbon barrier effect. But for farmed seaweed, it's totally different. It has a very strong carbon barrier effect. When we do some field survey, we can easily get a long sediment core, very rich in organic carbon. Based on a paper published recently, they report that the organic carbon in soft sediment below the seaweed farm can reach as high as 140 tons of carbon each hectare. And also there is a very high barrier effect. It is reported the organic carbon barrier rate averages nearly 2 tons CO2 equivalent each hectare each year in farm sediment. Our laboratory has been engaged in the research of seaweed farming carbon sequestration 10 years ago. Here I want to give a brief summary of the different processes driving carbon sequestration in the seaweed farming area. It mainly includes 5 processes. The sedimentary carbon sink we have mentioned earlier, and also it includes the dissolved organic carbon sequestration, suspended organic carbon sequestration, inorganic carbon sequestration, as well as the export of microcarbon to outside and to the deep sea. Later, I will introduce the detailed mechanisms or detailed processes. As we know, the microalgae will release a large amount of organic carbon outside. These include the dissolved organic carbon and particular organic carbon. Actually, the long-term carbon sequestration mainly depends on how much this seaweed released organic carbon can be permanently locked in the ocean. We know that the released organic carbon will be subject to microbial degradation. Once the organic carbon was transformed into a igneous form or recalcitrant form, driven by the well-known MCP process, it can persist in the ocean for a long time, as the RDUC can persist in the ocean for more than thousands or for hundreds to thousands of years. So this including the RDUC and RPUC. So regarding the carbon sequestration mechanism or the carbon sequestration effectiveness, we raise several important questions. The first question we want to ask, how much of the DUC released by seaweed can persist in the ocean as RDUC? During our field investigation, we found that in the seaweed farming area, despite there is a very strong water current and the water has a very short residence time, the concentration in the seaweed farming area is much higher than the surrounding areas. That indicates seaweed are a significant source of DOC release, thereby supporting a higher microbial abundance. What we want to know most importantly, most eagerly is how much of this released DOC can persist in the ocean. We know that the DOC will subject to microbial degradation. Therefore, we extracted the DOC from the seaweed farming area and conducted a long-term microbial degradation experiment for 1 year degradation. Finally, we found that in the first 2 months, the labile DOC can be easily or rapidly utilized by the microorganisms. And after that, the remaining DOC keeps very stable and show a very strong resistance to microbial degradation. Also, we conducted some photodegradation experiment. Finally, we find that about 58% of the DOC remained considered as RDOC. So this indicates that the seaweed farming has a significant contribution to dissolved organic carbon. And further study, we reveal that this RDOC come from both the contribution of the MCP and the macroalgae themselves. Due to time limitation, I will not introduce in more details. And we draw a figure to show this process clearly. And this paper has been published in ESD. The title is Carbon Sequestration in the Form of RDOC in Seaweed Farming Environment. So we have answered the ratio of RDOC in the seaweed farming environment. Most importantly, we want to know what is the total amount of RDOC that the kelp can contribute throughout their entire growth cycle. We know that the kelp grows at sea for nearly 200 days. It is challenging for us to conduct daily sampling and the daily monitoring. So we conducted several in situ mesocosm experiments during different growth stages, and we measured the daily contribution of RDOC. And by combining using the Gumpers model and the growth model, we roughly calculate the total RDOC contribution by kelp. As a result, and it is surprising to us, we found that the total RDOC contributed by kelp throughout their growth cycle is nearly equal to the carbon content in the harvest kelp biomass. This indicates neglecting RDOC would lead to serious underestimation of seaweed farms' contribution to ocean carbon sequestration. This is poorly understood in the past. And this paper has been published as a cover article in the Journal of Science China and Earth Sciences. The third question we want to ask, we know that large amount of organic carbon will be exported outside, but it is still an important knowledge gap. How much or how about the outward transport of seaweed-derived RDOC to the deeper sea? Since the RDOC is very stable, it has the precondition to conduct to undergo a long-distance transport. Previously, the literature just reported the POC export, just like this paper is published last year. In that paper, they mentioned the export of carbon, but also just refer to the POC. In that paper, they also mentioned their estimate does not include dissolved and refractory carbon pathways, and it highlights a relevant potential contribution of seaweed to natural carbon sinks. So why it is different? As I mentioned earlier, since DOC is very difficult to be measured using and the sources are very difficult to differentiate using isotope tracing or using other eDNA method, since it cannot extract its DNA. So now it is a very important knowledge gap. To address this question, we consider if we can establish the specific molecular fingerprints of the carp DOC, and then we trace their outward transport. Since the ultra resolution, high resolution mass spectrometry technique, such as the FT-RCR-MS, can identify the molecular species of a very complex DOC composition. And this can give us technical support. So firstly, we collected all the DOC during the carp growth and during the carp degradation. We get more than 5,000 carp-derived molecule formulas. And this fingerprint has removed the seawater background. And we know that although we get the carp-specific molecular fingerprint, since we think that there must be some overlap between different sources. We know that the phytoplankton and other macroalgae also release diverse DOC molecules. There must be some overlap. So we try our best to establish a most higher carp-specific molecular fingerprint. So we conduct a multi-step filter processes. We collect all the available data published, and we conducted many experiments. We collected the DOC molecular fingerprint from diverse other algae. This include 8 different phytoplankton types, including diatom and dinoflagellate, and also including some microalgae, including red algae, green algae. So after remove all the background noise from that other sources, we establish a highly carp-specific DOC fingerprint contains less than 2,000 DOC molecule formulas. And using the machine learning method, we differentiate the carp-LDOC and the carp-RDOC, especially based on their composition divergence. We developed a molecular inner score and help us to differentiate the export of carp-derived RDOC. And also, we use our carp-specific DOC fingerprint. We match against a global DOC dataset. This dataset covering both surface and deep ocean at different depths. Very interesting, we find that the carp-RDOC signatures were detected throughout global oceans, as far as 10,000 kilometers from the kelp ecosystem and can reach the deep ocean at depths of 6,000 meters. And most interesting, we found that the kelp RDOC molecule species have some overlap with the deep ocean DOC pool. And the kelp RDOC molecule comprise almost 5% of the total deep ocean DOC pool in molecular species. This is unexpected. So we also give a rough estimation that the microalgae system may contribute a RDOC production of nearly 150 t-gram carbon each year. And this paper has been submitted and is now in revision. The title is Global Disposal of Micro-Derived Recalcitrant DOC Contribute Significantly to Deep Ocean Carbon Storage. Now I have introduced the DOC. Another released organic carbon is POC. So we want to ask, what is the sequestration potential of the suspended POC released from growing seaweed? So due to time limitation, I will not introduce in details. Using the same method as described above, we extracted the POC released from the seaweed and conduct a long-term microdegradation. We found that the majority of POC released is readily degraded by microorganisms. After long-term degradation and photodegradation, a significant portion can be transformed in recalcitrant forms, enabling long-term carbon sequestration. And this sequestered fraction consists of 22% of RPOC and 14% RDOC. But it is still unknown how much POC released by kelp throughout its cycle ultimately contribute to carbon sequestration. This is the total amount. It is still awaiting further study. Here I want to emphasize a very interesting phenomenon we found. We found that the susceptibility of RPOC and RDOC to photodegradation differs dramatically. The RDOC is more susceptible to photodegradation. Its degradation rate is 10 times faster than RDOC. So then I want to ask the final question. Does seaweed contribute to inorganic carbon sequestration? Previous studies have reported that ecosystem like mangroves and salt marsh can contribute to inorganic carbon sequestration, mainly by increasing alkalinity. We know that total alkalinity is a key measure of seawater's capacity to store dissolved inorganic carbon. It enables DIC to remain stably stored as bicarbonate and carbonate ions, preventing it from readily escaping into the atmosphere as CO2. During our field service of large-scale macroalgae blooms, it is our proliferous green tide, we found that there is a significant increase of alkalinity in seawater. And our laboratory simulation experiment further confirmed that half of the generated DIC and nearly all total alkalinity remain stable in seawater across a range of conditions for a long time. And the enhancement of total alkalinity allowed more DIC to persist in seawater instead of escaping into the atmosphere, thereby function as a mechanism for inorganic carbon sequestration. We know that after the microalgae blooms or at the decline stage, a large amount of microalgae will sink into the seabed. So we also want to know what's the fate and the carbon sequestration potential of the sunk microalgae in coastal oceans. Interestingly, we found that after 2 years simulated microbial degradation, nearly 38% of the carbon in sunk macroalgae biomass were ultimately sequestered in various forms. Most interestingly, among the remaining carbon, as high as 10% existed as dissolved in organic bicarbonate ions. That indicates that it enhances the seawater alkalinity and contribute to in organic carbon sequestration. And we give this process a vivid name. It is microbially-driven alkalinity pump. We know that we reveal that this process are mainly driven by microbial process, mainly dominated by sulfate reduction and denitrification process. Now we have introduced all the carbon sequestration mechanisms. Now let's move on to another question. What's the significant potential of global seaweed for carbon sequestration? Based on the published papers, the literatures, and by combining our own estimations, finally, we give a rough estimate. The global carbon sequestration from seaweed is about 300 tgram carbon each year. This amount is tremendous. It is a very large contribution to carbon sequestration. And today's topic is seaweed farming. We know that China has the world's largest seaweed farming industry. In recent 2 decades, the seaweed farming industry grew very fast in China. We also give a very rough estimation. We found the annual carbon sequestration by seaweed farming in China now is over 5 million tons. It is measured in CO2. And the CO2 removal by seaweed farming, it is important, the potential. The seaweed farming has a large-scale implication potential to remove CO2, as it still has enormous room for development in terms of both area and economic output. Since seaweed farming area within the 40-meter isobats in coastal areas now account for only less than 0.3%. So it's a large room to conduct seaweed farming. And on the other hand, seaweed farming has multiple environmental benefits, including they can improve the water quality and can improve can mitigate the ocean acidification and the oxygenation, and also can increase the fishery diversity. So, from this perspective, seaweed farming has a great potential for carbon sequestration. However, we also do some related research. Under climate change scenarios, although it is very important to have great potential for seaweed farming, but the sustainability of seaweed farming is also threatened by changing climate and environmental conditions. In 2022, in Shandong province, a large-scale and severe carp mortality event caused a total loss of carp harvest in the seaweed farming area. We do some related research. We find due to extreme weather conditions and through a complex physical complex processes, leading to elevated seawater transparency, and finally cause the dysbiosis in kelp microbiome, then cause a large-scale mortality of kelp. And also by metagenomic analysis, we predict ocean warming and acidification are expected to accelerate kelp microbiome dysbiosis and mortality. Therefore, the sustainability of seaweed farming need our attention. Take-home message is that seaweed farming has significant potential to contribute ocean carbon sequestration and climate change mitigation. And this carbon sequestration capacity has been seriously underestimated due to the neglect of invisible carbon pathways, such as the production of RDOC and RPOC, and the enhancement of total alkalinity, as well as the export to deep ocean. And the sustainability of seaweed farming faces growing challenges from the effect of future climate change. That's all. Many thanks for your attention. And here, I also want to express my sincere thanks to the members of our laboratory for their contribution to this research. Thank you.
All right. Thank you so much, Yongyu.
Yeah, thank you.
An excellent and also elegant presentation on today's topic, because until today, I was not so convinced about how seaweed may help mitigate climate change at such scale you described. So thank you so much for giving us this opportunity to learn about seaweed as a way to mitigate climate change. Now, let's welcome Professor Gao from Xiamen University, Professor Jiang from Yellow Sea Fisheries Research Institute, and Professor Xiao from Zhejiang University. Yongyu, you welcome to stay to join the discussion. Professor Jiang, if you are here somewhere, I need to see you. Okay, good. Again, thank you all three for being on today's panel. Professor Jiang, I'll start with you because given your extensive and impactful work on integrated aquaculture, particularly your focus on seaweed bivalve co-production, could you elaborate on the ecological and economical significance of co-cultivating kelp and oysters, for example?
Thank you, Professor Zhang. Can you hear me?
Yes.
Okay. Good afternoon or morning, everybody. I'm Zengjie from Yongyu Zhang Research Institute. It's my great honor to be invited as the panel member of this topic. Just now, Professor Zhang Yongyu shared us a very excellent presentation and clarified the various forms of carbon migration and transformation processes and the mechanism driven by seaweed farming. From a scientific perspective, the carbon sink driven by seaweed farming belong to the category of carbon sink fisheries, and this concept was proposed by Professor Tang Qisheng, academician of Chinese Academy of Engineering in the year of 2010. Different with other types of carbon sinks, the carbon sink function of seaweed is driven by farming activities. In this context, I have two comments on this topic. The first one is that when we talk about the carbon sink function of the seaweed, actually we should pay attention to think about the sustainability of the farming activity. Here, I want to share one question with you. The question is that, are the more seaweed farmed, the more carbon sink will be? Of course, the answer is no. As we all know, there is an old saying called everything has both advantages and disadvantages, just as a coin has two sides. The unscientific farming activity can have a negative impact on the environment. So the scientificity of the farming activity is the key point in the realization of the carbon sink function. In this case, the theoretical support is the concept of agricultural carrying capacity. This concept is defined as the ability of the environment to accommodate a particular activity without an acceptable impact. And the second one is that, just as Professor Zhang Yongyu mentioned, seaweed is the important contributor of marine culture production in China, and the farming area is normally located within 20 meters, or some, he said, is 40 meters water depth. In this area, seaweed is not the only farming species, but also the shellfish, especially the bivalves with farm. They share the same narrow area together. The photosynthesis of the seaweed relies on three major factors: light, nutrient, and CO2. So the proper ratio of seaweed and bivalves biomass will maintain the balance of the carbon and oxygen in the ecosystem. For example, the seaweed can benefit from the dissolved nitrogen and phosphorus, and also the CO2 released by the respiration and calcification process of the bivalves. At the same time, seaweed can provide oxygen for bivalves. By this way, this positive interaction between seaweed and bivalves can not only generate more carbon sinks, but also achieve better economic benefits. The fishermen will be very happy because we can get more money, and we will have greater motivation to practice the farming activity. Finally, it forms a very good closed cycle. This is just my personal opinion and it's for reference only. Thank you.
Okay. All right. Thank you. Excellent comments on this topic and very insightful points. Now, let's turn to you, Professor Gao. Your pioneering research on algae-based carbon capture, utilization, and storage has garnered international recognition. Could you share with us your perspective on the role of blue carbon ecosystems in climate change mitigation from your perspective? Yongyu has already described us the scale that green kelp can help reducing CO2. What's your perspective?
Okay, thank you, Professor Zhang, and for this question. Firstly, I would like to clarify that blue carbon ecosystem here include mangroves, salt marshes, seagrasses, and both micro and macroalgae. Among these blue carbon plants, I think macroalgae stand out for one key reason. That is macroalgae, also called seaweed, can be found almost everywhere across the sunlit surface ocean. So the aerial resource is orders of larger than any other coastal vegetation alone. And in a recent paper, we showed that there is a sufficient area for seaweed cultivation to sequester 4 gigaton CO2 per year. The number is the required CO2 removal to stay below 2 degree warming based on RPC 2.6. And the second point is the economy, because as we know, one major challenge for CCS is the CO2 capture and storage is high cost, right? And so in terms of macroalgae, there is already multiple billion markets for food, feed, and pharmaceuticals from seaweeds. So if we direct a fraction of seaweed biomass to the existing seaweed market and using the remainders for CO2 long-term CO2 storage, it will be economically feasible CCUS rather than a pure cost burden, right? So Of course, the seaweed cultivation, particularly offshore seaweed cultivation, have to overcome the nutrient limitation environments because there are very few nutrients in oligotrophic oceans. And there are also two solutions for this problem. One is artificial upwelling system that can deliver the deep nutrient-rich seawater to the surface to fertilize the seaweed. And the second one is like Professor Jiang mentioned, is IMTA, integrated multi-trophic agriculture. So the waste from like fish and shellfish can fertilize the seaweed and support seaweed growth. In addition, I think for the offshore seaweed cultivation, the selection of suitable seaweed species is very important in different areas.
Thank you so much, Professor Gao, for that wonderful overview on the capture carbon in your perspective. Next, we'll have Professor Xiao from Zhejiang University. Professor Xiao, with your distinguished work in ecosystem health and resilience, and your acclaimed contributions to aquatic ecological restoration, how do you assess the potential of aquaculture systems, especially large-scale operations, to contribute meaningfully to carbon sequestration?
Okay, thank you, Professor Zhang. It's a very good question. As an emerging blue carbon ecosystem, seaweed demonstrated a significant carbon sequestration capacity, while also contributing to mitigation of ocean acidification and eutrophication. So as Professor Yongyu has just mentioned, recently we have tested the hypothesis that the seaweed farming aids in carbon burial. I can add some quick fact to that study, which is measured across 20 different seaweed farms all around the world. These farms ranged in age from 2 to 300 years and spanned in size from 1 to 15,000 hectares. So it's quite international, and the average organic carbon burial rate in this sediment beneath the seaweed farms was 1.87 tons carbon dioxide per hectare per year. So it's quite a powerful number. We can learn from our recent study. Additionally, we have also analyzed the changes in pH and carbon system parameters in surface waters of 3 seaweed farms across China. So comparing them with the surrounding seawater conditions, the findings indicated that the seaweed farming is effectively buffers acidification. So like the kelp, because it has the highest production, it has the highest buffering capacity, increasing pH by 0.1 units within the aquaculture area. It's also a very large number if you considering the decrease of pH in the past 100 years. So it's just equal to that scale. Furthermore, with China experiencing significant coastal eutrophication, we also quantified the nitrogen footprint and phosphorus footprint of Chinese seaweed farms. So the number is 1 hectare of seaweed farming could remove the nutrient loads to those entering 17.8 hectares for nitrogen and 126.7 hectares for phosphorus in coastal China. So all these different 3 evidence showing that the emerging seaweed blue carbon is very important, not only based on its effect of carbon removal and global change mitigation capacity, but also, as you mentioned, based on its contributions to ecosystem health and resilience. So that's all for the question. Thank you.
Okay. All right. Thank you so much, Professor Xiao, for your valuable insights on this topic. I would like to ask another round of a general question to our speaker as well as to our panel members. That is the tools you use. Although you all work in the general area of agriculture in terms of CO2 sequestration, resilience, or carbon capture, I'm sure you use perhaps different technologies. What is the most critical, effective tools in your field? I'll start with Yongyu, because you are now linking the CAP field with very high resolution mass spectrometer, FT-RCI-MS. How do you see the value of linking the CALP visual versus some very sophisticated technology to understand the invisible biomarkers to judge the importance of seaweed in CO2 mitigation?
This is a very good, excellent question. And now it is also a knowledge gap. We know that the carb-derived DOC is very complex, contains thousands of even different molecules. It's very super complex.
Right.
Now the current method is difficult to identify the detailed composition. The ultra high spectrometry method, such as the FT-RCI-MS, can differentiate the molecular species. So your question is how we establish, I think, the highly carb-specific molecular fingerprints. So we know that it is difficult. So we try our best to remove all the background noise from other producers, such as phytoplankton in seawater and other diverse microalgae, including red algae and green algae. Remove all the noise and then dissect only the very high specific molecule. It's a small amount of molecules. And then using these molecules, it is high specific. They trace the widespread of the carb-derived DOC molecule in the global oceans from the surface to the deep ocean. Finally, we found that the carb-derived RDOC molecules can reach the deepest ocean at 5,000, nearly 6,000 meters depth, and the covalent, the ratio is very high. It's nearly 5%. So using this method, we answered the question you asked. I'm not sure if I answered it well.
Well, that's very good. Thank you, Yongyu. So we'll start with Professor Gao again. What's your favorite tool? What's the most effective tool you think for your research?
Yeah, well, I think as other scientists, actually, nowadays we employ multiple methods to address one specific research question, because each method allows to understand this question from different dimensions. For me, here I would like highlight the mesocosm method, because we have some experience in conducting mesocosm experiments in Xiamen Bay and in Dongshan, Ireland as well. And so the advantage of a mesocosm experiment is it can bridge the critical gap between the lab work and the real world environment. Like Professor Yongyu Zhang said, the real world is very complicated. So you use a mesocosm, actually we can control the conditions, like we regulated the PCO2 under the temperature, so we can mimic the ocean acidification and ocean warming, and also because it's a large culture, and for our macrocosm, it is 4,000 liters seawater, so it can hold the entire ecosystem, and you can use
4,000
different
liters macrocosm?
Yes, 4,000 liters. Yeah, macrocosm, and
That's huge.
yes. So it's a it's kind of a real world, but it's not real world, but it's much larger than the labor system.
I see.
So I think yeah, it's a powerful tool for our group to investigate the effect of climate change on seaweed and on the ecosystem. Yeah.
Okay, excellent. Good luck in your macrocosm, 4,000 liter macrocosm. Okay.
Yeah, but it's not easy to run it. Yeah, actually.
Right. Okay, now let's go to Professor Jiang. What is the best way to study the interactions between seaweeds and bivalves, like oysters? How do you make them to grow happily together?
Yeah, actually, the ecosystem model is the key methodology what we use to identify the interaction among organisms and non-biological elements in the marine culture system. This model mainly covers three submodels. The first one is the hydrodynamic model, because as we all know, the farming activity is normally located in the open or semi-open area. Therefore, the water exchange process caused by the hydrodynamics is very important. And the second submodel is the what we call, and Yongyu also mentioned, is the individual group model. This model describes the growth variation of the seaweed or bivalves over time. The main theory currently relied upon is what we call the hydrodynamic energy budget theory, and this model provides a basic module for carrying capacity assessment and production prediction. The third submodel is the biogeochemical model. It mainly includes the nitrogen, phosphorus, silicon, and also phytoplankton, zooplankton, etc. Then, based on this coupled three submodels, we can get a lot of information, such as the final production and the ecosystem response, and also the economic benefit. And thereby, we can obtain the sustainable farming density of seaweed or bivalves. That's all.
Okay.
Thank you.
All right, thank you. Now, Professor Xiao, what's your favorite tool in your research?
I think it's the integrated multi-omics approach, because
Okay.
my work aims to unreveal how microbes, viruses, and metabolic functions work together to drive the in blue carbon ecosystems. So primarily, we will use combining the viromics, microbiome, and metabolomics and reactionomics.
Okay.
It's a very important integrated method because it allows us to move from simply listing biological parts to understand their dynamic functions. It answers the fundamental questions like who is there, what are they doing, how fast are they doing it. For instance, microbiomics tells us who is there, and microbiolomics reveals the viral predators and regulators that are infecting or controlling the microbes. And omics allows us to know the consequences of how these interactions by identifying the small molecules produced. This is what they are doing. And finally, the reactionomics quantifies the rate of these processes. That's how fast we can track the flow of the key metabolic pathways. So in short, this multi-omics is like a decoder. So by using this integrated approach, we can move from observations to truly understanding the mechanisms that determine the stability and function of the blue carbon ecosystems. Thank you.
Okay, very good. I'd like to thank you all for giving such wonderful presentation and comments. I can really appreciate what I learned today from very fundamental food as a necessity to feed people, to elegant research questions, and for the big global meaningful CO2 mitigation work. Thank you all for this opportunity. I know my co-host Yernar has some questions or comments regarding agriculture in the Caspian Sea. Do you want to join in with your question, Yernar?
Thank you, Professor Chuanlun Zhang, for giving me the privilege to co-organize this seminar series. I represent the Caspian Environmental Consortium, so I am using every chance to ask our speakers a question from the perspective of the Caspian Sea. I will explain why. At the last seminar, Professor Philip Boyd of University of Tasmania shared his view that the Caspian Sea and the Black Sea can be natural analogs to fast forward the assessment of marine CO2 removal via ocean alkalinity enhancement. I believe your excellent research on the carbon sequestration potential of macroalgae is also crucial for the Caspian Sea. So I would love to hear your point of view. And if it's possible, I would also like to hear about the potential impact of fisheries and fish aquaculture on the Caspian Sea. Thank you very much.
Okay, Yongyu, do you know much about seaweed production in the Caspian Sea?
Yeah, so thank you for your question. It's a very good question. From my perspective, I think carbon sequestration by seaweed farming is promising in this area. We know that the seaweed farming is not just for carbon sequestration. It itself is an ocean industry. The seaweed has a high economic value, can be used as food and materials and fertilizers. So it has a large-scale implication potential. From this perspective, I think it is promising. Yeah, but for the fisheries, I'm not in this area. I think this question should be answered by Zengjie, Professor Jiang, Zengjie. Yeah.
Right. Yeah, Zengjie, what you have any comments on making the Caspian Sea a good agriculture place?
Yes, I will try to answer the question because for the carbon sink fisheries, the definition is normally limited to the unfed agriculture. I'm not sure in that area what kind of fish agriculture, and this it need to input some food or something else. If so, maybe it's difficult to quantify its carbon sink function. Yeah.
Right. So, Yernar, you probably need to convince your government to develop a need for seaweed, for bivalves, for oysters.
Yeah.
Enhance those production, and at the same time to take care of CO2 from the air.
Yeah, because the food for the bivalves or even the seaweed, it is from the natural sea, from the phytoplankton or nutrients. But for the especially for the fish agriculture, they need food. They need some more. Yeah.
Well, I think we have come to the end of today's seminar. Thank you all so much for this again very valuable, very meaningful, impactful presentation and panel discussion. Thank you all very much. Okay, bye.
Bye.
Thank you very much.
Thank
Nice
you.
to see you. Thank you. Bye.